Welcome to the Blueprint of Life video 21. In this video, we're going to be discussing biotechnology, genetic engineering, and how we can produce transgenic species. So some of the words which I just mentioned, so biotechnology, any technique that uses living organisms to make products. It's an artificial process in which we manipulate the DNA of living organisms. Genetic engineering, where we genetically modify an organism by adding a desirable gene to the DNA, and then we can add or we can remove and substitute a gene to ensure that this is passed down to offspring. And then we can also have transgenic organisms when organisms' normal genome has been altered because this gene that has been selected, this transgene, has been introduced and it's from another species. So in terms of some applications, we can make genetically modified foods with increased nutrients, higher yields, and which can be processed more easily. They usually have we can introduce resistance to disease, pests and pesticides. We can treat disease, or it can be used as a reproductive technology as it increases the breeding success of individuals. So here's a diagram which pictorially represents what happens, but um, we insert a gene into a plasmid, which is extracted from a bacterium. We insert the gene in which we're interested in. It's put back into the bacterium. We can clone that, take the copies of that gene, and then introduce it into the organism. Here is a sequence of steps. So we cut the cut process is first. So the gene that is favorable is removed from a cell of an organism using restriction enzymes. We copy that that bacterium, that plasmid which has had the gene inserted into it. And then we paste it so the genes are inserted back into the egg cell of another species. Which after it's fertilized is now a part of the new organism's DNA. And then step 4, the egg develops into a mature organism, which we refer to a transgenic species. And then it's got that gene, which we manipulated in the first step, switched on and allows it to function. So in terms of how we can deliver that gene into an organism, we can have the micro injection of DNA directly into the nucleus of a cell. We have a process called biolistics, which we can deliver DNA mechanically onto microscopic particles in target tissues, and they're fired into those areas by a gene gun. We can have electroporation, where there's an electrical current which increases the permeability of the membrane, which allows the gene to move in, or we can have transduction by a viral vector, where the DNA is carried on this vector, are carried by vectors, including these viruses or plasmids. And a process which this happens in is when we use things like nasal spray or asthma puffers. The way in which we can recognize whether this gene has been successfully inserted or incorporated into an organism is that we can use what we call fluorescent markers, and they will fluoresce when that gene is turned on, which indicates to scientists in the laboratory that that gene has been successfully incorporated. One example which I'll discuss, and then you'll need to find a second example in class for the research task, is what we call Bt cotton, and that was produced in the 1990s in collaboration with some American scientists and the CSIRO. And the reason it's called Bt cotton is that a gene from Bacillus thuringiensis was incorporated into a cotton species, and that gene coded for the production of a protein, and that protein was important as it allowed the caterpillar of the Heliocervophasia moth to be killed when that protein was ingested by the caterpillar. Because that cotton now possessed that gene, it reduced the need for cotton farmers to kill these caterpillars with pesticides, which is better for the environment and reduce the development of pesticide resistance in lots of other bacteria and organisms. Um, the protein which was produced only kills the caterpillar when it's ingested, not humans, and not most animals and most insects, which makes it safe for the majority of the population. And it allowed cotton growers to change the pesticides that they were using. So now they only had to use narrow spectrum pesticides, which doesn't wipe out beneficial insects for the environment. Here we have a picture and some steps in which the process of BT cotton weight was produced. So they got a normal cotton seedling. There was a fragmented seedling which was infected with the BT genes. This fragment grew. The callus that grew um, had grew in a medium where the hormones produced an embryo. The cotton seedling produced from the embryo, and then we see that the new cotton seedling and that new mature plant is in fact a transgenic species with that BT cotton gene, incorporated into its genetics. The dot point also requires us to talk about some implications for society, and there are a number of ethical issues which arise. So we talk about those being in terms of the environment, in terms of financial and social justice issues, in terms of medical and health issues, in terms of animal and human rights, and then we talk about just is it the right thing to do? So here we have a quick summary of the points for and against. So I guess in that environment and nature, is it ethical to interfere with natural processes? And that's one of the against arguments. But also the for argument is that most new things are not always seen as the right thing to do until we understand them and understand that they are a benefit to society. And therefore, sometimes it's considered unethical if we don't use the science that we know to make society a better place. We talk about when we increase the yield or the drought tolerance or the resistance in plants, we talk about it being more cost effective for farmers. So they're able to have more of a financial gain. Uh, and then that can be put into further research. But then we talk about, well, can people afford it, and how do we then give ownership to different companies when they produce these species, and is that passed on to the consumer? And then again, we go down to medical and health and human rights issues, which you can look at in more detail. That's video 21. Make sure you're in class to complete the secondary research activities, and then make sure you tune in for video 22.